Colon Cancer and the Microbiome by Brooke Garza. Background. Colon cancer is a disease where cells within the colon grow uncontrollably, which then forms a tumor. Its disease state is categorized by stages. Stage 0 equals precancerous cells in the innermost lining of the colon to stage 4. Cancer has spread to distant organs. The impact of gut microbiome in colorectal cancer has risen significantly. Dietary molecules linked to colon cancer include short-chain fatty acids produced by gut bacteria fermenting fiber and beta-hydroxybutyrate, which is a ketone produced in a ketogenic diet. Epidemiological data shows a large increase in colon cancer, increased rates in young adults being diagnosed, and dietary links: high processed foods, red meats, and sugar. Some form of treatment include, but are not limited to, surgery, chemotherapy, radiation, and immunotherapy. Healthy microbiota. Gut microbiota is a diverse and unique community of microorganisms, mainly bacteria, that plays a vital role in maintaining overall health. Contains hundreds of different species of bacteria and yeast, rich in lactobacillus and bifidobacterium, which produces short-chain fatty acids, helps with immune system support by helping train and fight off harmful pathogens, and ferments carbohydrates that the body cannot digest on its own. Disease microbiota. Change in gut microbiome composition, known as dysbiosis, has been associated with an increased risk of colorectal cancer. Bacterial species such as Fusobacterium reclidum, Bacteroides fragilis, and Escherichia coli have been associated with tumor growth and inflammation. Altering microbiome can contribute to colon cancer by creating chronic inflammation, producing harmful toxins, and disrupting normal cell function. Some bacteria produce toxins like B. fragilis toxin, which can damage the gut lining. Mechanism. Microbes contribute to colon cancer by causing chronic inflammation, damaging DNA, and interfering with cell functions. This is achieved by invading the mucus layer, producing toxins such as coleobactin and other genotoxins, and creating metabolites that induce mutations/promote cell proliferation. An imbalance called dysbiosis allows procancerous bacteria to flourish, which leads to increased cell proliferation that then leads to tumor growth and progression. Experiment. The testing done shows the impact of gut microbes on the colon epithelial cell genome. Experiment. Bacterial presence resulting in numerous changes in histone acetylation and methylation in the proximal colon tissue of germ-free mice. Bacteria studied: murine gut microbiome, control group, cell-free mice, and mice. Method of testing: electrospray ionization tandem mass spectrometry. And the duration was 2015 to 2019. Future studies. To further our knowledge on colon cancer and the microbiome, I believe future studies should focus on developing microbiome-based testing for early detection, as well as gaining more insight on how the microbiome influences cell progression and treatment response. It would also be helpful if physicians would educate their patients on how to see the signs before it's too late. Summary. The gut microbiome is linked to colon cancer through many mechanisms, including the production of DNA-damaging toxins by certain bacteria, promotion of inflammation that fuels tumor growth, and protective metabolites like short-chain fatty acids. Dysbiosis can increase the risk of cancer, while a healthy microbiome can provide protection. Gut microbiome influences the effectiveness of colon treatments like immunotherapy. The article I used was Impact of the Gut Microbiome on the Genome and Epigenome of Colon Epithelial Cells, Contributions to Colorectal Cancer Development. It was published February 25th of 2019. There were multiple authors, and the source was Genome Medicine. Thank you for listening.